You're listening to Treating Blood Cancers, the LLS podcast series for professionals. We will be joined by cancer experts to discuss blood cancer diagnosis, treatment, side effects management, and the importance of clinical trials. They will share their experience in treating patients and discuss strategies for optimal patient care. Let's get the conversation started.
Welcome to Treating Blood Cancers, the LLS podcast series for professionals. I'm Dr. Ken Miller. I'm a medical oncologist, a hematologist, and very proud to be an LLS volunteer. And I want to thank all of you for joining us on this episode. Today, we're going to be discussing biomarker testing and the role of the healthcare team, including integrating biomarker testing into clinical practice and decision-making, insights into effective patient communication about biomarker testing, and future advancements and research in this field. We're delighted to be joined by Dr. Jennifer Rushton from Clinical Pathology Associates and the medical director of the Pathology and Laboratory Medicine Department at the Baptist Health System, and Dr. Eric Severson, who's Executive Director of Medical Affairs and Evidence Generation at LabCorp Oncology. Jennifer and Eric, thanks for joining us.
Thank you for having us, Ken.
Thanks for having us.
I want to start out with what are biomarkers from a very broad point of view, and then I'd love to hear some examples. Eric, tell us a little bit.
From a broad point of view, biomarkers are anything that is a characteristic of the tumor that we look at to try and understand either the diagnosis, the prognosis, or the appropriate treatment for the patient. This takes a large number of forms, whether it's things on the surface of the cell, so various proteins that you can look at through different methods. It could be specific changes in the DNA, so it can be various mutations. It can be things like changes in the chromosome, so large scale alterations there. Specific examples would be things like a BCR-ABL fusion that you find in chronic myelogenous leukemia, for example. There's a really wide array of different types of biomarkers, and it's really this catch-all term for everything that we're trying to identify about a patient's tumor.
Thank you. Excellent, because we throw the term around quite a bit. Jennifer, a question for you. For the practicing hematologist and oncologist who is a generalist like myself, I mean, there's a lot of blood cancers and there's a lot of biomarkers. What's your perspective in terms of how much are we, meaning clinicians and also pathologists like yourself, able to keep up? And how do we keep up? Any advice?
It's challenging. I think the best thing we can do is talk to each other. You may hear about new biomarkers, especially on the therapeutic side, that I may not know about yet, where I'm focusing more on the diagnostic side. I may know about a new immunohistochemical stain that's available, for example. So I think the best thing we can do is communicate with each other. Of course, we've got our resources. I love the WHO, that's the hematopathology main resource, but they only publish that every few years. It's really hard to keep up with the constant deluge of literature. I rely
Yep.
on our reference labs, like people like Eric, to let us know what kind of new biomarkers they're working on or that they're testing. And relatively recently, the pharmaceutical companies have also had medical liaisons that come out and reach out to pathologists, and I'm sure hematologists alike, letting us know what they're working on from a therapeutic side that may have a companion immunohistochemical stain, for example, that we might be able to test that might predict whether their new drug that they're working on will work on a patient. So, communication with all of these players, I think, is helpful.
And similar to Jennifer, I have sort of my favorite resources that I go to. NCCN has really detailed treatment guidelines, but in there they also have how do you do prognosis and diagnosis and what biomarker testing is appropriate in those settings. I find that to be a nice high-level summary resource that gets updated reasonably frequently. But then also working in a reference lab, we're kind of in a unique position where we work with physicians like Jennifer, where she'll ask questions about a new biomarker, and that's one way I'll learn about things. I'm like, oh, wait, people are asking about this. I need to go learn myself. We also interact with pharma partners quite a bit for things like day one readiness programs. So when a new companion diagnostic is going to be launched and they expect approval from the FDA, they work hand in hand with reference labs. So we have that test ready to go when the approval is announced, as opposed to once the approval is announced, okay, well, now we need 60 days or 3 months to
Yes,
validate
yes,
and launch the test.
right.
So we typically know about these things ahead of time so that we're ready when things start. And then we do educational initiatives to try and help people in the field understand what the new markers are and why they're important and how they should be used.
I really want to endorse what you were saying, Jennifer, about communicating, especially as the complexity has gone up in using biomarkers and in molecular diagnosis. Not only do I depend upon the hematopathologist, but I really enjoy the interaction because just exactly what you're saying, both of you, that there's an opportunity to share what I know, which has a certain focus to it, and vice versa to learn what you guys know as well. When I was a fellow early in my career, I worked with a hematologist who was part of the original FAB group, French American British group. Essentially, AML was diagnosed subtypes based on pretty much morphology. What is the role of morphology now? And then just looking back, based on morphology, would you say that the calls that we were making diagnoses back then were, likely to be 80% correct, 90%, 40%, et cetera? So, Jennifer, let me ask you again those questions about morphology. And then, Eric, if you could tell us more about the FAB, for example, and accuracy.
Yeah, I think morphology is still the foundation of our diagnoses. I think if you look now, the morphologic correlates, we've matched those with certain genetic mutations that explain those morphologic correlates. So we were right on at, identifying what we were seeing, and now we can match up the various biomarkers that go along with that morphology. I think what biomarkers have done, however, is in samples that are suboptimal or inconclusive, that gives us that edge when we're not quite sure about the morphology. We can identify a biomarker, and that will get us to the right classification. But you really still have to start with the morphology. That's our starting point. And we've talked about the vast array of biomarkers. You can't possibly test all of them. So the morphology gives you your starting point, and then you can narrow down what biomarkers might be helpful in helping refine your differential diagnosis.
Thank you, Eric. How about looking at accuracy in retrospect?
I think things were pretty accurate in retrospect. There are always going to be edge cases where the molecular diagnostics are going to tell you something that you would have never expected from looking under the microscope. I think what has happened, instead of having these broader categories where AML used to be a pretty broad category, now it's much more narrow. So it's AML with this particular type of biomarker or genomic alteration. And so instead of these broad categories that we could assign things by morphology to, we now have the morphology and kind of immunophenotype gives us kind of the general category, and the molecular helps us narrow it down further and further and further. But as Jennifer said, you can tell some things, you can guess what the molecular alteration is going to be with a pretty high confidence based on the morphology. One thing I wanted to add about the morphology as well is that it's critical for making fast decisions. So acute leukemia, for example, is something where you have to figure out what to do pretty quickly, and you want to know what that patient's risk is. And so that morphology is really that first step where once you've done the peripheral smear or once you've done the bone marrow biopsy, you can tell a clinician this is probably leukemia within a couple hours and get the really high priority biomarkers done within 48 hours if you're looking at something like acute promyelocytic leukemia where treatment timing is really of the essence. So the morphology is that starting point, as Jennifer said, but it also is much faster than anything else as well to get that very first set of information.
Let me ask Jennifer about when do you believe we should incorporate biomarker testing, and what are you doing in your own practice?
It's really incorporated very early on. As soon as you're deciding your differential diagnosis after you've seen a patient and perhaps a CBC, clinical presentation, at that point, there's even some markers that you can do on peripheral blood before perhaps you even decide on a bone marrow biopsy. And that may lead you to decide whether a bone marrow biopsy is even necessary or not. So it's really from the very beginning. That makes me mention the clinical history that you've seen with the patient is so critical for the hematopathologist. So, in my practice, the pathologist is primarily responsible for ordering biomarker testing on clinical specimens, bone marrows, lymph node biopsies, but I rely on that clinical history and the clinical presentation to guide me along with that morphology to decide what I should order. And if there's any doubt, of course, I reach out to the HEMONC to get more information, what biomarker testing might be useful for them to help clarify their differential diagnosis. So it really has to be right up front, I think.
No, I'll share a story literally from today, and it sort of speaks to what Eric was talking about with getting an answer quickly. I did a bone marrow this morning on a patient. I wasn't really sure he had pancytopenia, and I got a call actually just before this saying it looks like he's got AML. So it's not waiting for flow and it's not waiting for cytogenetics. It's real time and it allows me to take the right actions. So thank you for that. I wanted to ask about just in general, what would you advocate for? Who should decide, let's say community or academic hematology and oncology? Who should decide on and order the biomarker testing?
From a community pathologist perspective, I really like to be the steward of the tissue. Now, this is once you've obtained a bone marrow biopsy or a lymph node biopsy, for example. I know what kind of specimen we have, I know the quality of that specimen. Hopefully, I've gotten a great clinical history from you to guide me when I'm ordering. But then I really like to advocate for the pathologist to drive that testing. It's in our hands. We can most quickly facilitate that testing. And again, if there's any doubt, we can reach out to our HEMONCs for guidance. But I like to take control of that and really advocate for pathologist driven testing.
Yeah, yeah. And by the way, I agree with you. And it's in every other type of testing, it typically is pathologist driven. Eric, what would you like to add?
Being from a reference lab, we get to see the arrangements that everybody has. And it certainly spans the gamut from the pathologists kind of determine what testing happened to the hemoncs order the testing sort of independently of pathology. Some places duplicate tests get ordered, once from pathology, once from oncology. But the places that do the best job are really the places where there is that communication between the pathologists and the hemoncs. And sometimes the orders come from one or the other, but it's when there's that communication that makes sure the right testing gets ordered, the right amount of testing gets ordered, and the right specimens get tested. There's nothing worse than sort of finding out at the end that the test you performed that didn't give you great results was not on the best sample, and then having to go back and test an entirely different sample because the pathologist wasn't in the loop, or having tests that weren't necessarily pertinent for what the physician thought the clinical diagnosis was because the oncologist wasn't in the loop. So it's really when you have that partnership between the two that works best. And then it's just whatever the institution or practices sort of ordering patterns have been set up to be.
All right. So for both of you, I told you about doing a bone marrow this morning. I wrote 60-year-old man with anemia. Now, truth be told, I probably could have said a lot more. So I'll ask either one of you to volunteer, but what would help you more? What kind of description, what details would further our partnership, clinician and pathologist? So let me ask Eric that, and then we'll get Jennifer's take on it.
I mean, starting out with that description, my assumption would be okay, I'm looking to see whether or not this patient has MDS. The critical things that would be useful to know would be like duration. Is this a man with 5 years of cytopenias? Or is this something that's just happened recently and he presented with symptoms? That would kind of give me an idea. I'm going to guess in this case, this particular individual probably presented rather acutely if they ended up with a diagnosis of AML.
Yes.
So that duration is helpful. And then if there are any specific symptoms or pertinent things in the workup, it would be useful to know if this is the very first thing that's being done or if it's an extensive workup, rule out MDS type thing.
Got it.
Yeah, I have to echo that. The chronicity would definitely be helpful. If you do have a differential diagnosis, that's always helpful so we can make sure and, exclude certain things. And then Eric alluded to it, any testing you've already done. I don't see that a patient has had NGS on peripheral blood, for example, and I'm starting from scratch without knowing those findings. And then I'll somehow find out later that they had a pertinent finding on NGS and that may have prompted the bone marrow biopsy. So, any previous findings that may have been outside of my institution would also be helpful to know.
Yeah. And by the way, thank you. I think this discussion, and I hope for the listeners too, it's a reminder of how important the history is and how much better I think you guys can probably do your job. Let me ask you about synopsis. We get this in, for example, in breast cancer, there's usually a synoptic report, but in blood cancers, sometimes I get these kind of results, sometimes I don't. Thinking broadly about blood cancers, what would make for a very good, an excellent synoptic report? And let's say, for example, on AML or myelodysplasia. What are your thoughts?
We actually do this. It's a service that we offer, but it comes at the end of the diagnostic workup. It's important to communicate to the physician the findings as they're finalized, but it's nice to have that synopsis at the end. And what we include are the morphologic findings. What were the findings by flow cytometry? So, what are those markers that are expressed typically on the surface, but also inside the protein? What are the findings from cytogenetics or from fish? So, what are those large scale chromosomal rearrangements? And then, what are the findings from any molecular studies that were done? And then combining all that together into this is a diagnosis of acute myeloid leukemia that presented in this patient's bone marrow with 40% blasts and an IDH1 mutation. So that at the end there would be the synopsis, but you want to make sure you give that at the end, but you're giving the provider the information every step as you go along the way, because it can take 14 days to 21 days for all of those components to be completed, but you certainly don't want to wait that long to get a synoptic report that has everything at the end.
Yeah. Jennifer, same question for myeloma, because again, there's a lot of data that ends up coming back. What would make for a very good synopsis of all that information?
It would be very similar. So, morphologic findings, immunohistochemical findings, flow cytometry findings. For myeloma, prognostic FISH panel would be added in there. It's not standard of care yet for us to do NGS on myelomas, although I have seen that available. We still basically stop at the FISH prognostic panel. So I think all of those, and then we don't do this, but I love your question because we do synoptics for every other tumor, but in our practice, we really haven't incorporated it for leukemias and lymphomas. But something else that would be nice to incorporate would be this serum findings or SPEP, UPEP findings for myeloma. That would really round out the synoptic. So you're giving me ideas to build a synoptic here.
Oh, good. Well, this is a collaboration, right? We're talking about. In a sense, as a clinician, I think guidelines are very, very helpful now. And in terms of looking at biomarkers and using them, what's your sense in terms of, because you're seeing a lot of clinicians and a lot of material, a lot of patients and diagnoses, but discrepancy or adherence between practice and guidelines from your experience?
It varies dependent upon disease state. I would say for hematologic malignancies, especially AML, adherence seems to be pretty good. Most patients are getting the testing that they need. It's so critical for all aspects of treatment that that's been taken up pretty well. I'd say conversely, on the solid tumor side, it's been a lot more difficult to make sure that patients get all the testing they need, especially when it comes to next generation sequencing and comprehensive genomic profiling. The uptake there has been a little bit slower, but by and large, for hematologic malignancies, and especially AML, I haven't seen a lot of workups that have been kind of woefully incomplete.
Yeah, same. I think what I have seen is a lag in perhaps discontinuing some testing that may not be necessary anymore. For example, FISH for MDS was very common before we had molecular analysis. And really,
Right.
well, even before molecular, I think the data showed that if you had a good karyotype, you didn't necessarily need fish. And I still see people just
Yeah.
tempted to order that, and it doesn't usually add additional information. So perhaps it's hard to let go of things that we've had previously. And then the other thing about guidelines is they can only be so specific. There's still the art of pathology. One pathologist may have a certain threshold for ordering testing, like NGS for MDS, for example. So I may see something that makes me suspect that, and it's subtle. And so I may order the NGS, whereas another pathologist might not. So again, that conversation and the clinical suspicion would help guide that, but there's not necessarily standardization when it comes to pathologist ordering practices either.
Yeah. Let me pose this one to Jennifer, but as you have perhaps had the experience of either reviewing all this with patients, clinicians, sometimes the patients are also relatives or neighbors or things like that. What are the words you would use to describe sort of, essentially biomarker results so that people understand?
I often talk about precision medicine. I think patients like the idea that you can tailor the treatment and the diagnosis to their particular disease rather than a one size fits all approach. So that's what biomarkers help us do
Yes.
is look at your disease, identify what specifically about it we might be able to target, and targeted drugs are becoming more common where we can target a specific mutation that your disease might have. So I think that appeals to patients. One thing when talking about patients, I think a challenge is uncertainty with certain results. So I recently had a patient who was also a physician who had a bone marrow biopsy done to rule out myeloma. The marrow was negative for myeloma, but it was sent for cytogenetics rather routinely. And on cytogenetics, we identified a biomarker that's typically seen in MDS. Now, this patient had no cytopenias. So, now what do we do with that? And so, this physician who I know called me up and said, what does this mean? And it was easy for him to understand the idea of an incidental finding and that we could just maybe monitor his CBC, see if he develops any cytopenias over time. This might be a very early marker of future MDS. But I can imagine, especially for a non physician, that uncertainty could be very anxiety provoking. So that's one of the challenges with this. I think identifying incidental findings or findings that don't necessarily match the clinical presentation or the morphology that we're seeing. What do we do with those?
Yeah, absolutely. Thank you. Eric, thoughts on that?
Yeah, similar to Jennifer's experience, it's trying to make it general instead of going straight down and talking about, okay, here's the gene, here's the marker, here's what it means. Talking more about that broader precision medicine term and why we're doing the type of testing we're doing. It helps to kind of start with what the end goal is, like identifying the right sort of treatment or identifying, if they have a better or a worse prognosis, phrasing that appropriately.
Yes.
It helps to talk about it in those terms instead of going all the way down into the very specifics, like I would if I was talking to a fellow physician. And
Yes.
then once you have the end sort of defined, then kind of going more and talking about what their specific findings mean.
Yeah, yeah, interesting. Yeah, it sort of frames it because otherwise, I think it's hard for people, it's hard for clinicians for that matter, to integrate the body of data that we have on each patient and to prioritize because not all findings are equal in terms of importance. I want to talk about the topic of financial toxicity because as we've added tests, they're expensive. Jennifer, let me pose this one to you, but do you get a sense, or Eric for that matter, what does a workup cost and how is that affecting patients?
Yeah, I think workups for leukemia and lymphoma in particular can be very pricey. We've talked about immunohistochemistry, flow cytometry, FISH, molecular analysis. So all of these add up to the thousands. I think most of this is standard of care and covered by insurance, but I have heard of situations where NGS, for example, certain molecular tests are not covered by insurance, and the patient may receive a several $1,000 bill. So that's certainly a consideration, especially in our community setting when we order these tests. Is it necessary? We have to anticipate that the patient may receive that bill, so we want to be absolutely sure that it's necessary for the diagnosis or prognosis for this patient.
Yeah, thank you.
And that's a specific concern that we hear from a lot of physicians when we're talking to them about our testing is they are very concerned, especially for those larger NGS panels, about what impact is that going to have on this patient that's already dealing with all these other bills. What most companies that do this type of testing try and do is for those larger panels, they'll have financial assistance programs. And so they want patients to reach out if they do get a bill or even before the testing's performed, sort of understand that patient's financial situation. And there are usually ways to defray some of that post insurance cost if there is any. But it definitely is not a cheap set of workups. And it's not getting any cheaper as we're able to better and better target therapies with better and better testing. But that does mean more testing and more complex testing.
I want to ask you about the future. We've talked about the present, but it's hard to imagine, at least for me, what will be the next generation of biomarker testing. What are your thoughts on that?
To some degree, it's going to depend on what the next generation of treatments are,
Yeah.
as that's what ends up driving a lot of biomarker testing to begin with. I think they sort of evolve in tandem. One thing that we've seen in the past in hematologic malignancies, but that I think is going to become more widespread, is minimal or molecular residual disease, or MRD.
Yes. Correct.
There's been sort of a history of it in multiple myeloma, for example, by looking at IgH rearrangements with flow cytometry and ALL. But I think we're going to start seeing MRD much more regularly in things like AML and other diseases as we're able to sequence more and sequence more deeply. I think MRD is really going to be something that becomes more and more common both in hematologic malignancies and in solid tumors as we're able to have some more of these objective measures and get further and further sensitivity. And really, MRD helps with, and I think what it'll help the most with is identifying sort of escalation and de escalation of therapy and ultimately prognosis. Beyond MRD, the other types of biomarkers I think are really going to depend on sort of what happens in the CAR T field and with these antibody drug conjugates. So these are both things that are being developed where you have to know what to target on the cells. So as more targets are identified, we're going to have more diagnostic testing that goes along with both of those new type of therapeutics that are emerging.
Yeah, and it's going to be exciting to see it. And Jennifer, let me ask you in terms of your practice, if you could sort of pose things that you're going to need to do a better job to the big organizations that are developing things, people like Eric, any thoughts in terms of what you would ask for that sort of may prompt research to bring those things to you and to me too as clinicians?
Well, one thing that makes this testing more widely available is turning a molecular marker into an immunohistochemical stain, for example. We've identified a mutation, and not everyone has access to the molecular testing, or it takes a while. So let's transition that into an immunohistochemical stain, perhaps, so we can all bring into our labs, or even if we can't bring it in, we can send it to LabCorp. But eventually, perhaps we can bring it in house and have that same day rapid testing rather than having to send that out. So we've seen that a little bit now, but I think that would be really useful for the community setting or resource poor areas.
Got it. Thank you. So I want to say this has been a very interesting and enjoyable discussion. I think one of my takeaways, beside really getting some interesting thoughts about the future, but truly for today, how important it is really this collaboration between the lab and the pathologist and the clinician and the patient for that matter. So you have my promise for your colleagues. I'm going to start writing better history. And I want to take the opportunity, with all that said, I want to thank Dr. Jennifer Rushton from Clinical Pathology Associates, Medical Director of the Pathology Lab for the Baptist Health System, and Dr. Eric Severson, Executive Director of Medical Affairs and Evidence Generation at LabCorp. Jennifer, Eric, thanks for joining us.
Thanks so much.
Thank you so much, Ken.
I want to thank all of you for listening to this really terrific episode. For a listing of all of our healthcare professional continuing education activities, podcasts, and healthcare professional resources, including a fact sheet on biomarker testing, please visit lls.org/CE. For any questions or refer a patient to LLS, please contact our Information Resource Center by calling 800-955-4572. Information specialists provide personalized one-on-one support to help patients learn about their disease, treatment, financial, and support resources. LLS also provides other resources for patients, cancer survivors, and their families, including a series of podcasts that can be found at lls.org/support. I want to encourage you to sign up to receive notification of future podcast episodes by subscribing at treatingbloodcancers.org. And we look forward to you joining us on future episodes.
Thanks for listening to Treating Blood Cancers, the LLS podcast series for professionals. We can be found on iTunes and other podcatchers. You can subscribe at www.treatingbloodcancers.org and provide your suggestions for future topics. Visit our archives section on our website for other great podcasts. Be sure to rate and review us on iTunes. Keep up with LLS by following us on Twitter at LLSUSA and on Facebook at the Leukemia and Lymphoma Society, and access our professional continuing education activities by visiting lls.org/ce. Let's keep the conversation going. Until next time.